Parkinson's is a long-term neurodegenerative disease affecting the nerve cells in the brain and the motor system. Most people with Parkinson's start to develop symptoms when they're over 50, although around 1 in 20 people first experience symptoms when they're under 40. According to NHS, about 1 in 500 people are affected by Parkinson's disease, with males being more susceptible than females. An estimate of 127,000 people are affected in the UK alone. Today we will focus on how mitochondrial misregulation can lead to an early onset form of the disease. The pathway that we'll look at today involves the kinase PINK1, its substrate parkin, reactive oxygen species, ubiquitin, organelle, mitochondrion, and a lysosome. Mitochondria are the powerhouses of the cell and are a vital part of cell metabolism, generating energy in the form of ATP. Through this metabolic activity, free radicals are often generated, and these are very unstable molecules. Therefore, the cell has mechanisms in place to eat up damaged mitochondria by autophagy and recycle their contents. PINK1 is a protein kinase involved in this process. Upon mitochondrial stress or damage, PINK1 gets activated. This means that it can now add phosphates to its target substrates. And one of these substrates is parkin, an E3 ubiquitin ligase. When parkin gets phosphorylated by PINK1, it gets activated and can go and ubiquinate proteins on the outer mitochondrial membrane and tag them for degradation by the lysosome. In Parkinson's disease, PINK1 kinase is not active. Therefore, damaged mitochondria cannot be properly degraded. As a result, free radicals are not removed, and they can react with important cellular components such as DNA, cell membranes, proteins, and organelles, leading to premature neuronal loss and eventual neurodegeneration. A study conducted at the University of Dundee at the MRC-PPU focused on the effect of niclosamide and its analogues, AM85, AM86, and AM87 in the activation of PINK1. The researchers used an antibody which was specifically bound to activated parkin. They first expressed untagged parkin in both wild-type and PINK1 knockout cancer cells. These cells were then treated with different concentrations of niclosamide and analyzed using immunoblotting assays. A higher amount of activated parkin was expressed as niclosamide concentration increased, while inactive parkin was found in the PINK1 knockout cells. They then used a control and treated the rest of the samples with niclosamide, AM85, AM86, and AM87 respectively. The results showed that in the presence of niclosamide and AM85, the amount of activated parkin increased, revealing higher PINK1 activity. Further in vitro studies with primary neurons treated with niclosamide and AM85 showed an increase of parkin levels with neuronal growth. However, niclosamide and AM85's line of action in activating PINK1 isn't direct. Therefore, further in vivo studies need to be conducted, with researchers looking into direct activation approaches to treat Parkinson's disease. We spoke with lead investigator Professor Miatul Mukik about the impact of his study.
The niclosamide itself, or agents like niclosamide, are likely to be therapeutically attractive for Parkinson's. However, I think it does show, though, that you can take a compound that previously has been given to humans and it can activate an enzyme important in Parkinson's. It has done, it's led to a lot of interest into targeting PINK1 with regard to finding compounds that directly engage with PINK1 and activate it.